Hi dear students, welcome back to one more session of microbiology, and today we're going to talk about the concept of biogenesis. Previously we had mentioned about the theory of spontaneous generation, which mainly explained that life originated from non-living matter. And we had also discussed various experiments which were supporting as well as which were against the theory of spontaneous generation. So let's see what is the concept of biogenesis. The term biogenesis can be split into two: bio meaning life and genesis meaning beginning. And this concept was being put forward by Rudolf Virchow in 1858. And this was basically a hypothesis which he told earlier, which he told in 1858. And according to this, life originated from existing cells. That was what he told. And it was just a hypothesis, but he could not experimentally prove it. And later on, this concept led forward towards the cell theory. And the cell theory, as we all know, if you go to consider all organisms, they are mainly consist of one or more cells, and the cells are all they are the mainly the smallest living things. That was the thing which was considered to be. And the cells also exist from or also arise from the previously existing cells. So his concept was that all organisms are the descendants of the first cells. And now we know the modern cell theory. And if you go to talk about the modern cell theory is based upon the experiments which were extended over around 400 years. And it was not only like Virchow's experiments that formulated the cell theory. We have, the first cell was being discovered by Robert Hooke in about 1668, around that time. And later on, various other scientists like Sweden as well as Swan, Sweden and Swan, okay, they all did experiments on plants and animals. Now, if you go to consider Swan, he was working on plants, and Sweden was working on animals. And they put forward the first two concepts of the modern cell theory, stating that all organisms are made up of living cells, and cells form the basic units of life. These two concepts of the modern cell theory was put forward by Swan and Sweden. And after that came up the concept that cells arise from pre-existing cells, which was being put forward by Rudolf Virchow. So the concept of biogenesis is what? Life originates from pre-existing cells. That was being put forward by Rudolf Virchow. And the concept of the modern concept of cell theory, which means that all organisms are made up of cells, and the cells form the basic unit cells, as well as the concept of Rudolf Virchow has also been included in modern cell theory. And the credit of that is often being given, that is, the modern cell theory or the cell theory is in fact given, the credit of that has been given to Swan and Schleiden. So this was a theory of biogenesis. And even if the concept was being, I told you earlier, it was a concept which was hypothesized by Virchow, but he did not have any experimental proof to prove that life originated from pre-existing cells. So the Academy of Science in 1860, they put forward, they wanted to find a solution to it. And they put forward an award for that. That is to, and it mainly, my main purpose of it was to attempt by means of all well-designed experiments to cast new light on the question of the so-called spontaneous generations. That is, to put a death name to the theory of spontaneous generation. The Academy of Sciences put forward an award, and they wanted to find out who could lay forward lots of experiments so that the theory of spontaneous generation could be disapproved or not. And the award winner of this was Louis Pasteur, of course, by his efforts. And he confirmed the experiments of Reddy and Swan and all that, and he did a set of experiments called the filtration experiments. In that particular experiment, what he did was he kept a cotton in the sterile medium, okay, in the after the air has been filtered through it, and he found that, see, that is, if you have a cotton piece and you, if you take a particular media in a particular conical flask, and if you plug it with a cotton, okay, and keep it and of course the media in the conical flask was supposed to be sterile. So if you take a sterile medium in a conical flask and plug it with a cotton, what happens, the air that happens to enter into the conical flask also is being filtered through it, and so microbial growth was not being observed. So Pasteur, he came forward with the idea that microbes are being present in the dust but not in the air. Of course, the organisms which are growing in the conical flask uh, need air also, because most of them are being considered to be aerobic. So air will pass through the what? Through the cotton, but the dust cannot pass through it. So that's what he said, that microbes are being present in the dust but not in the air. And as a final proof, he did another experiment called the Swan neck flask experiment. What he did is, he placed nutrient solution in flasks, heated their necks in the flame, and drew them into a variety of curves called the Swan neck flasks. And he bent the neck of the flask in such a way that air can enter but the dust cannot. And he heated the broth and he found that there was no bacterial growth, and the broad neck of the flask was in such a way that it could permit the dust, but if you break the neck of the flask, okay, if you break this neck, but neck of this particular flask, what will happen, the dust will happen to enter into it, and then the growth will occur. So you can see a visual description of that particular, a visualization of that particular. This is the swan neck flask experiments of Louis Pasteur. Of course, this is Reddy's experiments, which was, I'm not discussing that here now. If you go to consider Louis Pasteur's experiment, first one, he took a flask which was not being covered, and open flask is allowed, air and any bacteria present in the air could enter. He could see that the flask is getting contaminated. And later on, he took another flask which was plugged with cotton, and the cotton plug filtered the bacteria from the air entering the flask, and the flask remained sterile. And finally, he did the swan neck flask experiment, where what is it? The, you know, the glass, if you heat it and you can extend it, you can form necks like this. So he heated it, and what does it means? The neck was being extended over here, and it is made in such a way that it was extended in such that the dust which happens to enter, or whatever it is, it will only reach till here, but it will not go to the side. It is in such a way, okay. So the bacteria were removed from the air entering the flask by settling in the long neck. So it was, if at all it enters, it will get down, or it will get settled over this long neck. In such a case also, what happens, the broth remains sterile over here. So this is the classical swan neck flask experiment of Louis Pasteur, which was enabling to disprove the spontaneous theory of generation. Now, the final death of spontaneous theory of generation was being put forward by the experiments which was being carried by Tyndall in 1877. He demonstrated that dust did indeed carry germs, and that if dust was absent, the broth remains sterile even if directly exposed to the air. Tyndall provided evidence for the existence of exceptionally heat-resistant forms of bacteria, and John discovered that the heat-resistant bacteria recognized by Tyndall, were species capable of producing bacterial endospores. So, with this, we come to an end of the theory of spontaneous generation, as well as the concept of biogenesis was being proved right now. it was being proposed by Virchow, but it was being proved, or the experimental proofs for the theory of biogenesis was being laid forward by Louis Pasteur. Hope so it is clear to you. With this, we come to an end of the session. Thank you.